Free your thoughts and watch them fly.
Welcome. Today we have with us a real treat, Origin of Life expert researchers, Dr. Roland Riek and Dr. Greenwald, who work together at ETH Zurich in Switzerland. It's a real honor to have you guys here. For those of you in the audience that maybe aren't familiar with your work, you are one of the world's leading experts, and your lab is one of the most prominent labs in researching the Amyloid World Hypothesis, amyloid chemistry, and the origins of life. So, Dr. Riek, it's a real honor to have you on the channel. Dr. Greenwald, thank you so much for coming on today. My very first question for you is, whenever you're talking with lay people just in your life, maybe at the supermarket or a friend of a friend, and you're trying to describe to them what it is that you do you have like a pre-canned elevator pitch about, what is your research? How do you describe it to a lay audience?
That's a brilliant idea. I should have a canned pitch. No, I always try to come up with something on the spot. It usually comes out really bad. But no, and I'm also have the difficulty that I often have to try to explain this in that German or Swiss German, which I'm not very good at. So I'm often like tongue-tied trying to explain. But no, but it is something it's something more to the point that I'm not really studying the origin of life. To me, that's a little bit too lofty to claim I'm doing. I'm trying to understand something that must happen before then. That's this origin of molecular complexity. How do you get molecules complex enough to where we could start picturing them doing things that look like life? I don't really watch a lot of videos on YouTube or follow a lot of stuff on social media, but I think you did a very good job. Actually, not a very good job. You did an excellent job of presenting the case of amyloids being involved in the origin of life. I mean, it's for someone who's not necessarily worked in the field, not necessarily so trivial, but I think you did well.
Well, thank you very much, Dr. Greenwald. I really appreciate you saying so. It's when I first came across the Amyloid World Hypothesis, I was just blown away by just how well it all lined up together. And I had, in my undergraduate studies, I have a biochemistry degree, and we went over RNA World just briefly in my biochem courses. And it was always one of the most interesting parts of the courses for me. I've always been fascinated with origin of life and abiogenesis, and just the complete novelty and freshness of the amyloid approach. I knew basically nothing about amyloids other than the buildup of amyloid plaque in the brain and all these negative connotations with prion diseases, medical issues, and just taking this whole category of biomolecules like amyloids and sort of rewriting their story, taking it away from these diseases, these, malfolded proteins, and all of a sudden showing the sort of integral role that amyloids could play at the origin of it all with life, and the kind of amazingly novel chemistry that is possible with amyloids. I mean, your work has been a big part of that. So what has that been like from your perspective to sort of how did you get into the field of amyloid research, and how has it been like for you to sort of watch this development?
Well, I have to say, it's all been kind of by accident. I didn't plan to join a lab where I could do this, but I like every probably thinking person, I've always wondered how life started. So it's the curiosity in me was already there, and I just got lucky enough to, through my sort of professional career, end up here in Switzerland at the ETH, where such things can be done, meaning you can kind of start working on a project that doesn't have some obvious connection to some application. So we didn't have really any direct funding for this in the beginning. It was just some institute type of funds that were able to support this. And then, since then, we have been able to get some funding, but as you, I think, mentioned in your first video, it's still rather difficult to just do pure origin of life research.
I have seen that that's been a recurring problem with origin of life research in general, is just the ability to secure funding. Even when I've spoken with, I've spoken about abiogenesis and spoken about this concept with other people before, and one of the first things that they'll ask is, so what kind of medical benefits would that have? What are the implications of discoveries? And I'm like, aren't you curious about how life originated? But I mean, I'm sure that there are lots of potent applications for amyloid research, but
right. So it's the kind of the case with all basic research. It can be done badly, but when it's done well, things come out of it that you can't predict, and that's the point to doing basic research. Is luckily we're a curious species, and oftentimes our curiosity uncovers novel things that we can have fun with or we can make use out of.
Exactly.
And oftentimes when you say, I'm going to directly try to solve this problem, you solve other problems along the way anyhow, or you don't even solve your problem because you were going about it all wrong, right? So you need to have these open-minded or open-ended type of research projects, I think, in any society to help the other more directed research have some foundation.
I always I enjoy reminding people about the story of the discovery of radio waves, where the discoverer was asked, oh, what are the applications for this amazing new discovery of yours? He's like, I couldn't actually think of any for radio waves. When you're talking with like lay people you might run into public that maybe don't have a science background, and they ask you, oh, what do you do for a living? Do you have an answer that you typically give them? And then could you maybe give us a brief overview of what the Amyloid World Hypothesis is?
So I have a long commute, so I'm sitting in the train working on my computer all the time, and some similar or the same people sit in the same time every week in the train, right? So they may ask, and they actually do, what are you doing here? What are these crazy things you are working on? And then I started to talk about science, and then I'm a professor at ETH, and then say, oh, great, what are you doing? And then I tell them just something new. Just must be something that nobody was thinking about. And then they say, why? Is that useful? I said, no, it's not useful. But it may be useful once, and we don't know yet, right? And here is one of these naive questions. Where is life beginning? I mean, we find small molecules and meteorites. There are amino acids. These are the building blocks of the proteins that we have. There are no nucleotides. There are small precursors, some small entities of nucleotides that the DNA is built up. That's what we start. And that's, of course, no life. This is molecules. And then the question is, can we go and make them more complex, that they are looking like a protein, that we have 25,000 of us in the body, different ones, billions of each of the copies usually, and they work there very hard to keep us alive. And can we make them more complex? And that is the start where we're thinking, how can we make out of these pieces of Lego? I'm not sure whether the Americans are also used to the Lego stones.
Oh, definitely.
So can we make the Legos more complicated, put them together, and then can they then make themselves so that they grow again? And that's something like life. Of course, it's by far not life yet. So that is the starting point. And what we found is that the simple, a protein has hundreds of these amino acids on a chain, and then they fold into a very complex structure, right? You may take your, so that's the chain here, and they fold up in a particular shape, and then they can do their function. But that's much too complicated. So we were thinking, can we make something small, very small, and then have another one, and another one, and another one, and they come together like a crystal, like a snow crystal, where on the top, actually like my fingers, and then they're suddenly forming a structure because they help each other. They get stabilized with each other. And indeed, this is possible. And the origin of that comes from Parkinson's disease, Alzheimer's disease, prion disease, or from your cooked egg white that gets white because small particles attach to each other and the light is not able to go through anymore, gets scattered. That's why when you cook egg white, these things unfold, these things here unfold and start to make these one-dimensional crystals we call amyloids. And that's where we started to look into that from a chemical and structural biology point of view. Discovered.
So, but coming back to why I'm on amyloids, as I say, I kind of got lucky where I landed, and also I joined the lab with Professor Roland Riek, who you'll probably also be talking to, and he has had a long history of working on amyloid fold and amyloids in general, starting from his graduate work. I think he worked on prion proteins, and then after that on other prions that do form amyloids as well. And so when I joined his lab, we were working on some of the functional prions and some of the disease-associated amyloids. And he had previous interactions with Leslie Orgel, Francis Crick at the Salk Institute, where actually he and I first met. I did my graduate studies in San Diego. And, he already had this spark of like, how could these small aggregates be involved in something besides diseases, right? Having functions, and in particular the origin of life. So that's kind of where the spark started. But it was, me joining Roland's lab that allowed me to then kind of pursue this. What we have shown is that, amyloids are inherently templating molecules. They template their own conformational transformation, like in prions, but also they can act as a template for their own chemical synthesis. And the interesting thing would be to see, since you can get a specific interaction between an RNA and an amyloid, can the RNA help template amino acids on an amyloid, right? Can influence what amino acids are being added, or vice versa, can an amyloid act as a template for RNA synthesis, right? And these are things that sound like, well, why should they work? But that's often, I can give you a hand-waving argument about why they would. But I think once experimentally we can show something like that is happening, it's rather compelling to say, oh, so these rather simple molecules can interact with each other and affect each other's synthesis. And that's also, a way you can eventually build up a system where RNA depends on protein and protein depends on RNA.
Also important maybe to realize is it's not so specific, that specificity is not so high in the requirement. So you may not need exactly the same sequence again to form an amyloid, but a close one. So it gives you, right? That means when you replicate, it may also work with something almost alike. And that is a strength in the early phase because specificity may be killing, because you cannot make that peptide anymore of the same composition.
So this would be touching on something I wanted to hear about, which was the heritability of these amyloid sequences. These amyloids can self-template and self-replicate in a way where they are adding amino acid residues to create a similar sequence as the template. So what you just pointed out there sounds analogous to a mutation. So it sounds like, and I'd like to hear from you on this, in terms of the propensity or capacity for amyloids to undergo a sort of Darwinian evolution. It sounds like, to me, the characteristics that would need to be in place are the ability to self-replicate, the ability to have some sort of primitive heritability, and then a variation in the population, which is exactly what you just described, how that variation can come about. So it sounds to me as if all of the properties are in place for amyloids to be capable of undergoing Darwinian evolution. Could you talk about that? And maybe is that a future goal of your lab to demonstrate in an experiment?
First of all, yes, it is something we want to do in the future, right? Evolution will be part of it, and adaptation to an environment, right? And then you get adaptation, and on the top of adaptation, you need some mutagenesis, and now not on the DNA, but on the peptide to work. I would like to highlight that in principle, we have a lot of knowledge from prion diseases, mad cow disease, Creutzfeldt-Jakob disease in this area. And they are called strains, like in viruses.
Wow.
And Charles Weissman showed 20 years ago, he had a cell line with prions, and then he could infect mice. And I don't remember now when, but the mice die at day 100 plus minus 10 days. So it's a very particular strain, 100 days they die. Then he added a drug to change the environment in the cell. And there it was about glycosylation pattern changing, and the prion protein has glycosylation, so it's a little bit a different molecule. But then there is another strain came up, and the mice died at day 300. So that strain was already there, but very little, right? And then when you take the way the drug, the old strain came up again, that all the mice died back at the 100 days plus minus 10 days or so. And then he could purify them down in terms of dilution experiments And once you had only one infectious prion strain entity left, that capability was lost. So with other words, you have even in the prions that which we have, right, in mad cow disease or Creutzfeldt-Jakob, you have multiple different conformations that are able to get more pronounced or less pronounced. It almost behaves like a virus, is able to adapt. And of course, this is on prions, and there's about strains. That means different polymorphic structures. It's always the same protein. Here we talk about, of course, we have a little bit different sequences and evolution that we think that should be possible indeed.
Wow, that's really amazing. Thank you for sharing that. The subject of Darwinian evolution applying to molecular systems has come up a couple times. And so I just wanted to touch on that because I hear pushback on that all the time from even people that are educated, saying that, oh, molecules are not capable of Darwinian evolution. That's only for already living things. And I try to explain, the way I think about it is if you have a population, whether that's living things or molecules, and you have a variation in fitness effects in that population, and for molecules, that could just be their stability, any kind of seepage pressure. And then if you have heritability to some degree or another, that's kind of the tricky one, right? But if you have those three things, to me, those are all of the necessary components for Darwinian evolution to take place. So can you talk about whether or not you can have Darwinian evolution for molecules or specifically amyloids, whether if they meet all of those criteria or if you disagree with my criteria and you have different ones?
Well, if you're defining Darwinian evolution as this selection of fitness through some sort of process where molecules compete for a resource or are able to replicate, then yeah, I think that for sure can happen. And whether it can be open-ended, like the definition of this NASA definition of life, which gets a little bit more difficult because it could be that you need a more complex system that looks already a bit like life in order for it to keep evolving, but that you can, molecules that are already able to some degree self-replicate and compete for resources, and I would say that already is showing that you can have such a system. I mean, you'll have other people who will poo-poo that and say, but that's not the same thing. But it's often just a question of semantics and how strict you want to say this, how like open-ended this type of evolution has to be. It might be rather boring, but it still fits the definition.
And then specifically in regard to amyloids and whether or not they have the chemical characteristics that would allow them to, in principle, undergo something like Darwinian evolution.
I think in principle, yes. And some of the missing experiments that we haven't been able to do yet because they're a bit tricky is to, instead of starting from our own pre-made amyloids that can do things like template themselves, and they can only template themselves kind of partly very well. The more you try to push that, the less robust they are. So that, what I'm basically saying is we start with a 7, 6, 7, 8 residue peptide. We add a shorter peptide to it, and we show that one peptide can act as a template to chemically finish off the rest. Similar to how RNA or DNA gets copied from a template and a primer. And what we would like to show is that you start with something that has no amyloids, because we know that they will spontaneously form under certain conditions. And then we want to look at what formed there, how it formed, and say, did that involve some sort of selection where certain molecules got made much more than others because not only they were more stable, but they were able to like to impose their own sequence on the rest of the pool. And that seems, we've shown mechanistically that could happen. To show that it is happening is a bit difficult. And just to put numbers on that briefly, if you want to take the system of amino acids, there's 20 of them that we currently have in biology. But even if you limit it to like 10 or so, right? If you want to talk about how many molecules can you make that have 10 amino acids that are 10 amino acids long? So let's say only 8. So this is for 10 amino acids, it's 10 to the power of 8, right? And that one I haven't memorized, but it's a large number. And this number gets very large very fast as soon as you start including more amino acids and more. And the numbers get so large that for a 100 amino acid peptide, which is not a big protein, life is full of ones that are thousands. And if you take all 20 amino acids, there's so many possibilities of 100 amino acid peptides that it's one of these, it's 20 to the 100th. And it's a number that is most people that are not mathematicians can't fathom. I mean, it's a number so big that you can't even make a single copy of each one of these molecules with all the known matter in the universe, right? There's not enough matter.
I have my doubts that even the mathematicians can fathom it. I think they're just telling us they can.
No, but I mean, it's sort of like outside the scope of, anyways, I got a bit off topic there. My point was that if I want to study a system that has very few, let's say only 10 amino acids, and only 8 residues long, and I want to look at all the peptides that are formed, the potential that are there is a very large number.
Yeah, the sequence space is immense.
Yeah, and then I want to try to fish out of there something interesting, and that's where technology I'm a little bit stuck with, but I don't doubt in a few years there'll be mass spectrometers and some other tools that will allow me to analyze this on a level that says, yes, there are potentially selections going on there that look like some sort of, you might call it Darwinian evolution. The problem is, right now I'm just talking about the initial synthesis, but I would then like to apply some pressure, like do that under this temperature, and show, oh, now this sequence is winning, now this sequence is winning, right?
Yeah.
Because their stabilities change. And that's something that I think is, totally feasible, like theoretically, but I just don't have the tools right now experimentally to do it.
I cannot wait to see what kind of publications come out of your lab in the future, Dr. Greenwald. I want to ask about where you see these amyloids forming. Is it primarily at hydrothermal vents deep in the oceans, or shallow seas, or maybe like an alkaline lake? What kind of environments do you see as being, fortuitous for amyloids to form?
I think the beauty of it is that amyloids are stable. You can put them into 50% ethanol or in 4 molar salt. I mean, very different environments, low pH, high pH, acidic, basic pH. So they are stable, but you need a certain concentration of it because it's a peptide that aggregates. So a certain concentration is needed. If you have a single peptide alone, nothing will happen. So from this, first, of course, you need synthesis of them, and you may need some activation surrounding. There might be some lava stones or a porous stone in a vent where you may get high local concentration. And what we do is we use COS gas, carbonyl sulfide gas, as activation agents. So you may need a little bit of reactivity around that. You get peptides one side, and the other thing is you need to concentrate. It could also be in a tide pool, right? Where the water evaporates and you get a high concentration, and then it may again gets diluted. Things like that could be possible. But NASA is also interested in them because you might still find them on Mars samples because they're so stable. We wait until the samples will be returned and then to be ready to measure. But
wow. Yeah, that's amazing. Just a highlight of the true stability of amyloids. I wanted to ask, it sounded like there's a range of different possibilities of environments for amyloids to form. They're so ubiquitous and stable. But it sounded like maybe the most probable, or at least the one that maybe ticked off the most boxes that you were going through in terms of like the availability of carbonyl sulfide and prebiotically relevant volcanic gas catalysts, compartmentalization, maybe like natural proton gradients, or all the things kind of seem to combine together at hydrothermal vents. Do you kind of consider those to be the most likely candidate, or?
You need reactivity or a dry wet cycle. And I think I like the vents. They look really like life could originate from them, right? When you look at the movie, but there are not so many of them, and they get not so old. That's what I read. They're only a few million years old, or even less. So that may be a counter argument, but I like that.
That leads perfectly into my next question, which was something that I get frequently when talking about Amyloid World Hypothesis. And let me know if you've got a better term than that. I recently got the Methods in Enzymology volume on peptide chemistry that you and Dr. Riek helped co-author a chapter on. And I noticed that Amyloid World Hypothesis was referred to by that term in that chapter. But there was one particular part that I wanted to highlight where at the very end of the introduction, it was noted that Amyloid World Hypothesis as such is not mutually exclusive to RNA World Hypothesis, and you can have RNA and amyloids arising independently or co-arising or interacting. And I just wanted to ask you a little bit about that, just to clarify that for the audience, and also to maybe talk about how maybe you see things as coming. Like, do you see the amyloids and the RNAs and maybe metabolic reactive systems all arising independently and then interacting, or like how do you see that all playing out, if at all you have a good sense of that?
Okay, so personally, I feel like metabolism is something that almost needs to be going before you can have too much of this talk of amyloid world and RNA world. It might not be the case, but what I mean by metabolism, I just mean some sort of chemical cycles that are taking some input of energy, some gradient of energy that can be eaten to create more complex molecules in this case. They're not necessarily going to be energy or food. So real metabolism is taking energy and converting it into food and other energy sources that can be used by the living system. Right now, I'm thinking of it more as a machine to increase molecular complexity or to select for, in some way, certain molecules to be created over others, right? So that they're just the ones that can be made faster, the ones that are more stable, or they can build cycles. That's the important part, where they help reinforce their own creation. So I feel like that something like that needs to kind of get started on the planet to create the building blocks, so there's enough of them in high enough concentrations to do the chemistry that we are publishing about, which is how do amyloids form, or how other people work on, how do nucleotides build long enough chains to make catalytic RNAs. Then it's, to me, obvious, though, that we can't say right now which one's really happening first. We can say that, amino acids are simpler than RNA. That's just a chemical situation, but maybe nature found a way of making those first. So the point is, there's no reason to exclude one system over the other. That's why we also say it's mutually, they can be, they're not mutually exclusive, but the opposite means they are mutually inclusive. Thank you.
Yeah.
And they have these molecules that we're interested in, peptides or amyloids and RNA, do interact in biology. So there's no reason to think that if they existed, they wouldn't have interacted in pre-biology. And so that's something we work on as well, just because we are curious about what are the possibilities of amyloids having specific interactions with RNA, and what does this do to both of those molecules. We've shown that it can stabilize them just through their interaction. Stabilizing, in this case, often means protecting themselves from hydrolysis, because that's like the big danger. And for RNA in a prebiotic world, it's you make it and then everything's water on the planet, possibly if it's in the ocean, and then it falls apart. So if it can bind to something, whether it be a membrane or a rock or an amyloid, it could protect itself from hydrolysis. And that's kind of the primitive state we're at right now. We haven't made life in the lab, despite what some you'll see plenty of times news stories that origin of life problem solved. It's typically an RNA story. But that's because those are splashy headlines and gets people to click on them. But the fact is, we're really in this very, very primitive stage where we're just looking, what are the potential players? What are their capabilities? What can they do? And they play well together.
A few questions off of that. The first one is, I know that your lab has done these experiments on the interactions of amyloids and RNA building blocks. I wanted to ask, have you extended that to looking at how, if nucleosides interact differently than nucleotides or nucleobases alone? Or I know that Dr. Sutherland's lab has been a big proponent of this cyanide-based prebiotic chemistry. Have you thought about, or have you conducted any experiments of along like how the precursors to RNA interact with amyloids, and if there's anything there.
Right. So we have tried to kind of understand or parameterize what are the requirements for amyloids to interact with nucleotides and polynucleotides. We are not specifically looking at synthetic routes like Sutherland has done to all of these molecules, and it's very fascinating work where they can show you can get all sorts of different types of prebiotic molecules from amino acids to sugars and nucleic bases, all in sort of not the same condition, but in a series of compatible conditions, right? No, so what we've done is we've looked at what are the compositional requirements of a nucleotide to interact with an amyloid, and on what are the compositional requirements of the amyloid. I mean, something that I feel like in our pre-discussion and what we've talked about so far is that we haven't really pointed out that the term amyloid is a little bit too generic. It's basically it's all that tells us is the structural state of the molecule. It tells us nothing about its properties, or they're just as varied as they can have as much variety as any other protein in principle, right? The idea behind the amyloid hypothesis is that some rather simple structure, this cross beta motif, can be the let's say the scaffold upon which a lot of these functions are built. But the point is, amyloids can be very different from each other. So we're not studying just one amyloid. We study lots of different peptides that can form amyloids or not, and try to understand what that implications that have for origin and molecular complexity. But then coming back to the question of RNA interaction, we look at different amyloids and see what are the characteristics of those amyloids that allow them to interact with RNA. And we look at the length of the RNA that's required down to a single nucleotide, down to a nucleoside, mononucleotide, monophosphate, this triphosphate, to understand the charge characteristics that are required, the sequence of the RNA. And it's not like we're getting some hard and fast rules, but it seems clear that the charge is playing a very big role. And that means that, if you have a nucleoside, so no phosphate, the interaction is significantly weaker. And a triphosphate binds much more than a monophosphate, and then a longer nucleotide, so a trinucleotide, 3 bases in a row, bind much more than a mononucleotide. And we have tried to understand what is the difference between the sequence of, for example, GGG in the RNA language compared to AAA binding or GAG binding. We do see that they bind differently, but it's not as simple as watching Crick base pairing explaining why they bind, because now we're binding RNA to a protein and we don't necessarily know the details. But the point is that we do see that you can have specificity on the base level as well as on the amino acid level.
We've touched on the fact that amyloids can act as catalysts for a myriad number of different chemical reactions. They have a wide range that they can catalyze. Can you maybe talk about specifically, maybe if there's any research that your lab is doing or look forward to doing in the future about, can amyloids catalyze any prebiotically, metabolically relevant reactions?
Yeah, I think since they can have catalytic activity, and has been shown by others that per weight, they're the most active amyloids, or most active peptide protein sequences. Sorry. So yeah, we think of that, and we there exploit the unique property of an amyloid. You have every 4.7 angstrom. That means 10 to the minus 10 meter, right? You have the same structure again, the same positive charge or the same negative charge. And so small molecules like ATP, adenosine triphosphate, have 3 negative charges. They bind well to an amyloid, while its analog ADP, which has lower energy, and ATP, of course, is our fuel in our body, doesn't bind to amyloid because only 2 negative charges. And this is like an avidity effect, right? That you have if you have 3, while you have 2, it's the activity binding is much less active. And you're often thinking from this perspective that So what we really would like to make is out of ADP, ATP, and this is the ATP synthase machinery that is so crucial in our body. Is in the mitochondria being the powerhouse, right?
Yes.
So making chemical fuel for us is the ATP.
Yes,
So
I think I've heard the fact that the average human creates their own body weight in
yeah,
ATP every single day. It's just
it,
recycled. So it seems like this really is at the very heart of metabolisms in basically all forms of life is this ATP chemistry that it sounds like amyloids have the or may have the capacity to catalyze.
yeah, and there has been some publication showing that it may be possible under that they are able to do the opposite hydrolysis, right, from ATP to ADP,
Mhm.
and an enzyme goes, of course, in both directions, and so. Of course, peptide amyls are not enzymes. Enzymes are very sophisticated machines, but the chemical reaction goes in both directions. So, yes, I mean, if you ask an RNA origin of life person, he would say, of course, ATP, this was just around, or ADP. I'm not so sure. I mean, you need something behind it. And if you think about ADP to ATP, that's one thing. You need to make ATP, but then what is the ATP doing when it binds to protein? It
Yeah.
changes the conformation of a protein from one to another state. Now, when ADP is not binding an amyloid, but ATP binds an amyloid, it stabilizes also an amyloid. So not only that you may catalyze with the amyloid help from ADP to ATP, but actually it already is a precursor of what the machineries are doing, namely stabilizing a certain conformation, and
Yeah.
then maybe it falls apart when ATP gets hydrolyzed to ADP. So these are the two things, again, which I think in early life or the prebiotic life, a lot of different roles need to be playing together, right?
Yes. And it also is interesting, right? Because I'd like to hear your thoughts about if you have this potential interaction between amyloid peptides and ATP, that's adenosine triphosphate. So adenosine is a nucleoside based on adenine, which is the A in the genetic code. Um, is there any way that this interaction with ATP can sort of lead to a more complex interactome of peptides and nucleic acids, like that may eventually become sort of a population of peptides and RNAs that kind of resembles tRNAs or eventually maybe the ribosome. Is that a sort of direction that you think is likely for an amyloid world to develop?
I agree with you. Now we talk about two things, right? Polymerase, I mean, synthesis of RNA or DNA molecules that could happen on an amyloid. And the opposite, translation, making proteins in the ribosome. And we are just at the moment looking into this, but this is streaming, right? We look at amyloid sequences in the ribosome, on the RNA, and on the protein, just as you ask. Or transfer RNA, which is maybe the precursor of the enzymatic active site on the ribosome.
Yeah.
As you speak, right? I have here the data and should work on this rather than give an interview. Just look at, do we find some motifs that might be interesting? It's maybe dreaming, right? I mean, this is yeah.
But you're getting me excited over here, which was, the ribosome seems like such an integral component for this entire process beginning from the genetic code, the dogma of biochemistry, the last universal common ancestor, all of it. And so the ribosome is fundamentally an interaction between proteins and RNA. Um, and so does this seem like a big major key in understanding how life arose and the origin of this entire abiogenic process is understanding where the ribosome came about? And is there any thinking in the field similar to how eukaryotes were like Lynn Margulis came up with the idea that eukaryotes arose from some sort of symbiotic relationship between two prokaryotes. Is there that kind of conception behind the evolution of the ribosome as arising of this synthetic interaction in this peptide amyloid protein world, that maybe you have these populations of self-replicating RNAs or self-replicating peptides, and it's the symbiosis between the two that leads to the ribosome and ultimately life? Is that at all in the background of any of this?
Sure. Well, I mean, I don't want to disappoint you, but you're like thinking on this as probably advanced as anybody else's, because it's just the type of thing that's very difficult to study, or maybe impossible to study. So you said it very well, that the fact that the ribosome is made of RNA and protein, and that its job is to read RNA and convert it into protein, has this like kind of knot that you need to try to untangle and understand where did that come from, and that it exists in every, you know, form of life that we know about. So it seems sort of obvious that, well, not obvious, it's a fact that RNA and protein started working together at some point early in life, and that's also why I don't like the idea of this RNA world hypothesis. I feel like they were together all along. But yeah, unfortunately, we won't answer those questions about when they came together, but that's going back to what I just talked about with our experiment, where we'd like to show how an amyloid at least could have been involved in an RNA interaction that was, you would never be able to draw all the steps out to how you went from there to what our ribosome looks like now. But the idea is to kind of piece steps together that you could logically think are happening in the course of history.
Do you have any kind of sense as to how far away we are from getting like an RNA protein system in a lab that maybe is capable of undergoing self-replication and Darwinian evolution, maybe of like together the peptides and RNAs in a population that maybe could resemble something like not like a ribosome, but maybe like some sort of precursor that maybe has a different function, or maybe a population of tRNA like molecules or something like that?
I'm not super optimistic that we'll create like stable living systems, because once again, that comes to like the situation that it's the stable living systems are not just like one life, but they're multiple things that all work together so that they could stabilize each other. And it wasn't just, you would say, one organism, but many things working together. And so I think we will create processes that look like possibly they're undergoing a lot of the things we talk about as life, but they'll be dependent, of course, on us providing all of what they need to, you know, in terms of energy and nutrients, and removing and doing a lot of the housekeeping for them, removing the bad stuff to create the environment that's stable for them. I mean, I like to think of the problem here. So, you know, other people approach life, or the understanding what's required for life, from this more top-down approach. And one of the approaches is you take a simple organism, you try to strip out as many genes as possible and find out what are the minimum number of genes that this can live on. And of course, doesn't mean that's the minimal number. It's just the minimal number based on the way life looks like right now. I forget if it's a few hundred. I used to know this, but it's on the order of, I think, a few hundred genes. But then the question is, what happens if you try to like, that's maybe, you know, there's a rather crippled bacteria that can survive for a while. What happens if you take a bacteria and you somehow kill it? You denature one of its proteins and it's just not working anymore. Can you take that bacteria and put the working protein back in there and sort of breathe life back into it? And as far as I know, the research on taking something that's really officially not living in some sense, not just suspended, and also talking about like a single bacterium and getting it to grow again is not trivial. There's a whole lot of processes that need to be working at the same time to create this. And that's just an example of, you know, life as we know it. You're talking about like life in a test tube, which would in principle be much simpler. And that's why I think that might be possible to create this thing that looks like life. But that's why the definitions are, you know, being fuzzy. Someone will say it is, and I will say, well, you know, it has some features of life. The one problem I have with trying to pin down definitions is that it narrows our mind down to looking at something that we want to call a system. you in your original video, you make the classic comparison about like, is it, you know, a virus, is it alive, is it living? And these questions of that are, to me, a little bit missing the point, because a virus is something, so a sign of life, and it only exists because of life, and it is then, in my mind, part of life, right? Whether I want to identify it as alive or not is not really so important. Just like, do I want to identify my skin cells as alive? They are, but they're part of a larger system, me, and am I alive without, you know, the bacteria who are not me? Yeah. No, I'm not. And also I'm dependent on all of the life around me. I don't mean other humans, but I mean like the fungus to the trees and everything to maintain an environment that I can live in, right? And so to then try to create life by calling just one system, it's almost like we're trying to create a living thing. What we need to create is a system in which we can call what's going on in their life, right? And I don't want to focus on whether something is alive or something not.
That's a really great approach. I mean, it's like missing the forest for the trees in a very literal sense.
Yeah. And so also I should preface, say that since I don't think it's ever going to get close in my lifetime to creating, life in the lab, I'm more trying to focus on something that maybe I can accomplish, which is understand something that I feel needs to happen, or almost certainly needs to happen before you have the things we might call life. And that's going from the molecules that we really have a strong feeling are going to be around in the universe before life, to the ones that you need to start to have life. And we don't know what that is, but so I'm looking at this origin of molecular complexity. How do you get from the molecules, that sort of we can make appear rather easily, to those that, like RNA, for example, or nucleotide. It's not clear where those come from, but we feel they need to be there before you have life, or something close to it. Maybe the life had a different nucleotide that evolved to RNA. Those are also, totally legitimate things, but you need to have more complexity than just alanine and phosphate before you have life. And so that my amyloid view of amyloids is that they help systems create this type of complexity through their different properties.
Because of this channel, I was able to get this book that you recently co-authored a chapter in, Methods of Enzymology. And I learned something new about amyloids and abiogenesis in reading your chapter, which was specifically, you brought up this point that as we look at more and more basal species, like on a phylogenetic tree, we see that their proteomes, that like the total amount of proteins in these cells, become more and more amyloidogenic. Like they basically tend towards amyloid folds more and more of a predisposition, right? Could you talk about that and what that implies about the origins of life?
So we have very, very few information about the early phase of life, right? Because it's so long time ago. And so one thing, of course, you could look into, what are the sequences, the protein sequences with the composition of the amino acids? What are they composed of? And so we took the entire genome information known, I think from 2020, and made small peptides of length 6 amino acids, or 7 or 8 or 9 amino acids, and just looked into all proteins through. Something interesting struck me the following. If you have 6 amino acids lengths, and you have at every single position have 20 different amino acid possibilities, because we assume you have 20 amino acids today. So we have so many possibilities. This means 20 times 20 times 20, so 20 times to the order of 6, that nature could evolve in it, right? The whole sequence space of a peptide length of 6, nature was able to screen. If you take, which is an average size protein of 200 amino acids, you need, I think, 10 or 10, you need many, many times more the universe in mass to make a single copy of each of them. So with length 9 already, nature was able to only screen 1% of a length of peptide 9, which is the typical amyloid length. So the sequence space that nature was able to screen in the 3 billion years was only 1% of all the other range. So now you can ask the question, is that one that amyloid sequence space up here that nature was not evolving smaller or bigger than the one that it was able to screen? And the one that was able to screen is higher than the random one around. And that would strongly indicate peptide amyloids were involved. They are functional, and they're still functional. And the more complex the organism was, in particular, if you go to the mammalians and the primates, the less of that amyloid sequences you find because they were less important. New protein species evolved. And nature could not randomize it. There's still information of the past because of that small sequence space that nature was able to screen so far.
Yes. So you mentioned something there about calculating the sequence spaces with the 20 amino acids that we have today. Is there any particular evidence or reason to go with this? Like, because I understand that you can form amyloids with basically just one amino acid up to 20, or, I don't know how many different possible amino acids are prebiotically relevant on Earth, but is there any reason not to think that the 20 amino acids that we have today arose out of a much more complicated soup of all kinds of different amino acid residue amyloids?
It could be more, it could be less. Of course, complicated amino acids came later because you need to make them probably, and it's more complicated to make them. But you can go down to a few amino acids which are rather simple. You find on meteorites already in a decent amount. And then already you can make a peptide amyloid. So there is, as you said, it is a primitive structure, if you so will, and so it needs only primitive precursors, these amino acids, and then it already works. It can also work with more complex amino acids. We have one life form on Earth, and all life originated from this, or only that one survived. This is the L amino acid life. This is, I talked to you before that the amino acids are composed of, the proteins are composed of amino acids, but there are two handiness, L and D amino acid, and you only find one of the two, right? You look in the mirror, and that's not you, and this life doesn't exist. There is, what we realize, there is very little possibility to detect the other life form because you cannot do PCR. You cannot feed, you cannot grow them. And the amino acids form exactly the same as peptide amyloids, whether they're L or D. They are chiral selective for one or the other. But both of them would be able to exist. And that's something I would like to look into, that deep abiogenetic environment, also because there's up to 15% amino acid content.
Very fascinating. And this actually touches on a question that I wanted to ask you later about the intersection of amyloids and homochirality. You often hear one of the big problems in abiogenesis is exactly what you brought up about how all life we have today is the left-handed, and then there's the right-handed chemical amino acids that can form at the same route, and they don't match. Exactly the problem you brought up. So the issue for any hypothesis of abiogenesis is to explain why life today is homochiral, all L amino acids. And so it sounds like Amyloid World has a very compelling answer to this question about how amyloids can selectively add on L amino acids onto their template versus D amino acids. Do you want to touch on homochirality and amyloids?
Yeah, but for that we would need a structure, and I don't have a structure model here. That model is not really a good model anymore. I don't think we can show that. The issue is that when you make peptide where you have several amino acids on top of each other, the side chains of the one handiness look into the one direction, look like that up. No,
Yeah.
look at in one direction. So if you make an amyloid, they all look in one direction or in the other one.
Yes.
If you would have another handiness, they would look into this direction where there is no space to, because the next one comes at it.
Mm.
And that's why you only can get poly L or poly D. Right? Either the L would be like the amino acids would be like that, and the D, the amino acids will look into this direction. So you can make again the D only, then it will work again, but you cannot have a mixture of the two. And so you get a selection. You may start with one or with the other one, but once you have it and they make themselves, you're stuck with the L or with the D.
I have to say that when I first learned about the Amyloid World Hypothesis, it was specifically this fact about the homochirality and how elegantly amyloid chemistry answers the homochirality question in abiogenesis. That's what really I found one of the most compelling parts of the hypothesis when I was first coming across it. I think the initial paper I read was CPJ Mori's 2018 review, and that was my initial introduction. And I was just really struck by the elegance of the homochirality solution, just explaining it with, like you just did with amyloid chemistry itself.
I mean, the elegance is already in the thinking of Murray, because he didn't do any experiment, right? Just by thinking about it's so elegant that it will work, it must work. And that's actually what happens in the lab, right? You can make it, almost every single experiment works because it's there, right? And that's what a theory usually indicates is a good theory.
What you were talking about with the membranes maintaining this disequilibrium across these gradients of with this divided by this membrane, this seems to be a really key aspect of life, this compartmentalization. And I've heard origin of life researchers saying, that's one of the reasons why we're looking at like a hydrothermal vent, for instance, because it has these naturally occurring proton gradients. It has naturally occurring compartmentalization in the argonite. And I think that it was the researcher that received the Nobel Prize for the discovery of proton gradients in the cell. And he actually had a really interesting conception about what life is, where I think maybe it's a little bit more similar to your conception of life, where he said life is not just this singular thing. It is a membrane from which one side of the membrane is kept at a disequilibrium from the other. And whether or not that membrane is our skin, our cell membranes, it's just that the living thing is the separation between the environment and the interior. And maintaining that barrier is what life is. And so that maybe could be seen as a beneficial way of motivating the approach of like exactly what you're describing as compartmentalizing the amyloids and your chemistry within these different compartments. Do you have any thoughts about that way of thinking about life?
Yeah. So, I mean, I think look at it a little bit differently in that the membrane is important for being able to convert some sort of energy gradient into another. So the proton gradient that exists across the membrane is a way of storing some potential that can be used later to whether to make ATP, etc., and or for signaling in nerve cells or something like that. But I think what probably one of the most important functions of the cell is to keep things from diluting too much. I mean, it has many functions, but if something gets an important interaction gets made between an RNA and a protein at one point, which we think happened, and they start to like work well together, if those guys could have wandered away too quickly, those special units, then unless there's something that can happen continuously over and over, that would have been kind of lost information. And they need to maintain that contact long enough for them to do something like reproduce themselves, for example. And I think that whether it's a membrane or some other sort of like a pore in the rock, there has to be some sort of containment, some compartmentalization early on in life, or abiogenesis.
Yeah, perfect. And that could also touch on when you were talking about the difference between a dead cell and a living cell, and what's the difference is maybe the concentration gradients across those membranes is what is so difficult about reanimating this dead cell, is getting those gradients back to the living state.
Right. Those at the right time, and also every protein in the right place in the cell, because it's not just that the proteins are there, but they have to be kind of ready to do what they're supposed to do and be, in the right location in the right concentration.
So would you say that it is a major goal of your lab to characterize amyloid's ability to catalyze metabolically relevant reactions?
So one of the first things we worked on when I got into the Amyloid World sort of hypothesis, even though I don't like the name, I do say it, getting into the idea of amyloids, is their catalytic capabilities, because that's sort of something that peptides are often not great at, as being catalysts if they're small enough, because they don't have enough sort of surface area to bind to molecules to create like a substrate binding site. And that's something I wanted to show. Coincidentally, at the same time, it became interesting for a lot of other people. So the competition got big enough in the amyloid catalysis field that I moved on to these other things like the templating and interaction with RNA and fatty acids, etc. And so it's still currently not our main point to try to recreate all of metabolism with amyloids. Right now, I'm personally interested in looking at what happens when you look in more complex systems, and that means taking, so whether it's RNA and or vesicle membranes, and seeing what that does to the environment of the amyloid, and can we create systems which are far enough from equilibrium to where they tend to keep change, and that you can look at some evolution and look how the energy coming in converts into something else in the system.
Wow, that is exactly what I had written down. Essentially, I wanted to ask you about a more complex system of amyloids, fatty acids, and nucleic acids all in the same flask. And I wanted to ask you specifically about whether or not that is something that you are pursuing with the goals of getting to a self-templating, self-replicating system that is capable of Darwinian evolution, and just letting that go for generation on generation. Is that like a main end goal for your research?
Sure. That would be an end goal, and that's sort of ambitious. So I would say that's a nice thing to like look down at the horizon, but really things have happened in much smaller steps than that. When you talk to Roland, you're going to get a little bit of a different, probably optimism, right? He's like a real, everything can be done, and we're like this close to solving problems. You'll notice the difference between how he and I talk. But I look at it not as like I'm going to get anything looking like life in a lab. I'm trying to understand the mechanisms by which things can go from one step to the next. And I have these little steps I like to work on. And sort of the next one is, how can you take, for example, a temperature gradient or a gradient in some energy source and convert it into a system that evolves. So for example, let's say we have, RNA doesn't get through a membrane too well because of its charge, but perhaps it can bind to a hydrophobic amyloid because it also has a positive charge on it, and that helps it get through the membrane, right? And then that would require some high concentration of RNA on the outside so that it wants to move into the inside. That's one type of gradient. And then you would have some system on the cell on the inside which would allow either that RNA to polymerize or act as a template for amyloid polymerization or something like that. And the idea is that, inside of a vesicle, so I'm talking about a fatty acid vesicle or a lipid vesicle, the environment is really different than on the outside. Both the pH can be different because of the nearness you are to the membrane compared to on the outside, but you're also protected from other external molecules. So you could have things happening on the inside that are different from the outside, and eventually it might be nice to see something like an amyloid that grows large enough inside the cell to create some stress which causes it to divide, something like that. These are all things that I can fantasize about, but and those are like the impetus behind us wanting to do some experiments, but we don't have those experiments yet, right? Those are the goals.
So I'll leave the the really in-depth science questions about amyloid chemistry there, and I want to actually touch on something that you brought up a little bit earlier when you brought up the RNA world people. And it seems like the origin of life field might be fragmented into sort of cliques of, maybe you have these RNA people that they don't want to hear about amyloids, right? Or maybe you have the amyloid people that think, oh, those RNA people. Is it cliquish? And if that is the case, do you feel that how do you feel that exception or traction of the amyloid world hypothesis, is it gaining in the field? Because I oftentimes will bring it up, even with biochemists, and there's not a recognition yet of, oh, amyloids aren't those disease-causing agents. It still seems like they maybe the last 15 years of amyloid research hasn't percolated into the field, and I'm curious to get your take on that. Do you think it's just it'll get there, it's just slowly happening, or do you think maybe is there a reason why, you know, what are some of the hesitancies that you pick up in the field?
I think if you are successful in a field, right, and with the RNA molecules, why to bother, right? Why to bother with something else? And people may get, oh, that's interesting. That's what we find, but so what? And that was like 10, 15 years ago. Jason just came back from a conference in the US and said, wow, everybody is so excited about the amyloids. So it takes sometimes time.
Yeah.
Of course, because you need to change all your chemistry, you need to change your thinking, and it's not so straightforward. We have no problems with the interaction of the RNA now. We got quite a few. We now understand amyloids rather okayish, and so we can now make that step and take the RNA on, and we see that the RNA binds tightly to the amyloid, which is a good step. The RNA stabilizes the amyloid stabilizes the RNA, and that sooner or later in life they came together, and the membrane also, right? Membrane composition,
Hmm.
and we should look into this level. And of course, the RNA is much too complicated to begin with. The ribosomes have so many nucleotides. The chances that this is so slim that there is absence of another helper that might be the peptide amyloid that could have evolved easily. But we also need to say the statistics. So what? We have one life form. May it just happened, right? We don't have a statistics. We have 100 different life forms, and 99 times it is the ribosome or the RNA that started, and one times the amyloid. We don't have a statistics, and so the statistical reasoning may not work, right?
Hmm.
Just
I wanted to actually ask you something similar to that of what do you like? So you've been in this field now for you're pretty established. I feel like you've probably heard a lot of, you know, scientists coming at you with criticisms of the amyloid world hypothesis. And so I have yet to hear those criticisms. I've been asking people, what are the criticisms that you can think of? I've been looking, and so far I haven't gotten any. So I'm actually curious, what are the top criticisms that you hear of the amyloid world hypothesis?
I don't think there is a that's interesting. They never criticize. They may only think, yeah, that's a side niche. Just go on with it. At the end of the day, of course, the strengths they always say is on the RNA side is that you have storage and catalytic activity, chemical activity on both sides, and you have the ribosome that makes the protein. So, and that's full of, on the catalytic side too, of RNA. But if you look at the ribosome, this is a complex structure, right? This is very, very late in evolution. And so peptide amyloids can store information and can be catalytic active as well, but much more simple. The biggest criticism I think I hear now when you start slowly thinking about is that the catalytic activity of an amyloid is little because it has a large surface which is water exposed. How you now make that, because a lot of chemistry needs to have somehow a control on the water environment. How is that working? I think this is the biggest criticism, and now we have already now a structure in our hands that actually will highlight this is not
Like fibrillar networks of amyloids can have hydrophobic pockets within them, correct?
exactly.
Yes, I think I've read a few of your work on that. So
And
a lot of
we have now high resolution structure almost finished, where we suddenly have hydrophobic pockets that are very hydrophobic. And so
I do have
that's
a question about that actually. Do you see those sort of fibrillar networks um, existing within or coexisting alongside of lipid membranes? Is there an interaction between these? Because the fibrillar networks seem very large to me. So how do those interact with lipid membranes if that's an area that has been looked into?
what you find is when you look at disease-associated amyloids that they touch a membrane, again, because of this repetitive structure, every 4.7 angstrom, lipids have a repetitive structure of maybe 1 angstrom, right? A little bit more tight. So you will see this interaction going on. And there is a cryo-EM structure of alpha-synuclein amyloid fibrils, which are associated with Parkinson's disease, that you see actually this membranes bound to the amyloid and highly organized fashion. So the interaction is present, and yeah.
Into a big question that I have, which is, for a hypothesis that has now been on the scene for, maybe 10 years, give or take, maybe a little bit longer. A lot of the times, even when I'm talking with professional biochemists, it still hasn't broken through. Like I'm still, if I say amyloid world hypothesis, a lot of people are still not recognizing that. I think it's only happened 1 or 2 times where I've spoken with somebody and they were already aware of it before I told them about it. So I just want to, I want to get your take about why you think that maybe it's still a little bit more obscure or it hasn't gone mainstream. Is it because there hasn't been that big discovery like a ribosome yet? Or why is it that you think that more people are not talking about this? Even like origin of life researchers like Jack Shostak or somebody like it seems like maybe these discoveries about amyloids and amyloids role in abiogenesis maybe hasn't percolated all the way through the scientific and public community yet. Why do you think that is?
All right, there's a couple things I want to say. First, I want to comment on the name Amyloid World Hypothesis. I think you wrote in the email to us, or I don't even know if you're okay with the term. I'm not okay with that term as like a reality, right? It's a useful phrase to sort of as a counterpoint to this, camp of the RNA world. But it makes no more sense, or it makes less sense than the RNA world in my mind, to talk about worlds where these molecules are the only thing or the most important thing that exists. But the other thing about why it's taking so long to catch hold, and maybe I'm not so impatient. I don't feel like it's taking so long. I mean, there was a lot of ideas that were, at least I'm aware of, in the early part of this century, where people were proposing amyloids could have some other functions. We were one of the first to start showing experimentally what some of these functions could be. Other people have. And amyloids have a huge interest for their material properties as well. So there's a whole research field going in that direction. But why is the general scientific community not so aware of the amyloid world hypothesis? I take that to be something that it's the type of subject you would learn in a textbook in college, first of all, and then from there maybe expand on it. And maybe in 10 years, this will be mentioned in more textbooks, or at least the RNA world hypothesis won't be presented so matter-of-factly, right? I mean, even I've watched over time, the Wikipedia page is sort of like evolved for amyloid world hypothesis to be like, yeah, there's a lot of, they presented more of a hypothesis and less of a fact, right? It's not so much like a lot of physical laws that we take them kind of for fact. It's really just a plain out hypothesis that has a lot of huge open questions.
When I learned about abiogenesis in my textbook in class, I think RNA world was the only one brought up. Not iron sulfur, not metabolism first,
exactly.
not first, just RNA.
And that was just, it was a fad, I would say, at the time. It was really popular. I don't know who wrote those textbooks and how those decisions were made. And I can't say they were political or personal, but decisions were made, and that sort of influenced what became popular.
And would you say that in the origin of life community, you have like certain camps, like you have the RNA people, and they're not going to go over and be open-minded about amyloids? Is there that kind of clique, like the clique?
Yeah, unfortunately, I think people tend to be a little bit that way. I don't know if that's a bigger problem, or probably the bigger problem is that as humans, we kind of are, and scientists, humans, we have our like hypothesis and we put a lot of effort into it. And so we want to believe that there's some validity or this is important. And so we put our blinders on, or we're not as open-minded as we should be. And I think it's partly that too. I don't think it's an intentional thing that people want to be in camps. And from my perspective too, over the years, as I presented my case in many sort of meetings and talked to people, the reception has gotten a little easier. People are much more aware of amyloids being functional, not necessarily being these like very very stable disease states that can never do anything, right? Mm-hmm. Think of them as more dynamic species, and it's something that just takes time to seep into the collective consciousness, if you will, of scientists.